Personalized medicine is changing everything, but turning potential into reality starts with one thing: access to reliable biomarkers, from promise to pressure. Thousands of clinical trials worldwide rely on biomarker-based diagnostics, and exosomes are quickly becoming a central focus. Once a niche research topic, exosomes are now among the top areas in biomedical science. The future of liquid biopsy is already here. Meet ExoGAG, the high-yield, fast, affordable exosome isolation system. Exosome isolation? It's now as easy as 1, 2, 3. First, spin your sample for 5 minutes to remove debris. Transfer the supernatant, then discard the pellet. Second, add ExoGAG reagent, twice the sample volume. Mix gently until solution turns blue. Incubate for 5 minutes at 4 degrees Celsius, then spin again for 15 minutes. Third, carefully discard the supernatant and resuspend the dark blue exosome pellet in buffer. The sample is now ready for the next steps. ExoGAG delivers high specificity, minimal sample, consistent results, works with your standard centrifuge, no need for specialized equipment, and it's fast. Use your isolated exosomes for downstream applications like biomarker discovery, diagnostics, therapeutic development, or drug delivery. ExoGAG is part of a growing portfolio with kits optimized for plasma, urine, saliva, CSF, and more. Precision science, made easy. ExoGAG, from Nexotech.